Is it like on campus or?
No, here on campus. No, no.
That's what it was like. You know, as much as I enjoy seeing grasshopper grows, I would rather like,
Grasshoppers don't bite. They don't bite. They might vomit on you, but they don't bite. Yeah. Yeah, so next week we'll be outside. We are counting ragweed on campus. We'll get spatial dispersion. Meantime, don't forget, we had the quiz that's live. Closes later tonight. We have our exams coming up in about a week and a half, I believe. Yeah, no, it would be a Friday. Is it this next Friday? Let's double check that. Yeah, today at noon, that's our halfway point. Is this it? Maybe. I wonder if this is this year's. No. Yeah, it's probably, I think next week Friday is our exam, so start reviewing. The exam will include this marker capture stuff, so estimating population size. We'll have to do that. In the meantime, let's continue our lecture. This is intraspecific variation. We've been talking about it. We haven't really addressed how do we quantify it. How do we separate the variation attributed to the genotype from the variation that's attributed to phenotypic plasticity? Right? That's what we're looking at. How do we distinguish between that? The classic way to do it is to perform this common garden experiment. This common garden experiment is to take individuals from one environment and grow them in the opposite environment, and vice versa. So it's kind of like a reciprocal transfer study, where you take individuals that were in these habitats, grow them over into the opposite environments. If you couldn't do that, then what you could do is take individuals that from these two different habitats and grow them up all in one place, in the same place. So you collect the plants from outside, you bring them to a central place, let's say at the greenhouse, where we use the same potting soil. We get the same light, the same watering regime, right? That's our common garden experiment. And then what you do is you see what happens to the phenotypes. So we know what the phenotypes are in their natural environment. Now what we want to do is see what are the phenotypes when they're raised in the common garden. If the differences in the phenotypes persist, then that's likely due to the genetics, to the genotypes. If, however, the phenotypes change, that part is our phenotypic plasticity. So let's kind of diagram this out, because I've got the example. All right, I'll leave this up here so you can kind of keep taking the notes. So what we're going to do is set up this experiment where we have a hill, and you have some plants that grow at the bottom of the hill and at the top of the hill. At the bottom, we generate tall plants. At the top of the hill, we have short plants. And our question is this due to the genetics, or is this due to phenotypic plasticity? So what we would do is raise those up in the same environment, but for us, what we're going to do is do more of a reciprocal transfer study. take these tall plants, collect their offspring, grow them up at the top of the hill, collect the short plant offsprings, transplant them down or grow them at the bottom of the hill and see what happens. So, we have our height. We have our two locations. At the bottom of the hill, our open boxes will be the tall plants. So these are the plants that were from the bottom. And our hatched will be those plants that were from the top, or at least the offspring. So we know if we take offspring from the bottom plants, grow them at the bottom, they'll be tall. If we take plants, offspring from those at the top of the hill and grow them at the top, they'll be short. So far, so good. What we can notice is maybe those bottom plants, when grown at the top, are shorter than if they were grown at the bottom. And those plants grown at the top, when they're transplanted and grown here at the bottom, they might be taller. So these changes from there to here, and from here to here, these differences, the change in our phenotypes, represent our phenotypic plasticity. That's the intraspecific variation that's due to being raised in the environment. Our plant was able to alter its phenotype to better match the environment in which it's grown in. Difference here between these two, so from there to there and there to there, the difference between our top and bottom plants, this represents our genetics. That's our genetic contribution. The tall plants still remain tall. The short plants are still short. Still have that tall versus short distinction. But the absolute heights were determined by our phenotypic plasticity. If, however, switch this. Here's our bottom, there's our top. If we get something like this, same type of graph, just different bars, different locations. Is there any phenotypic plasticity affecting this variation? Yes or no? No. Did the genotypes change? No. We took the bottom plants, when they grew them at the top, they were the same height. We took the top plants, grew them at the bottom, they were the same height. But we do have genetics. So this variation is all due to genetics. It's all due to genetics. And we could do one more. We're at the bottom, there's our top. we get something like that, what's producing the intraspecific variation? That's our phenotypic plasticity. Our genetics, we saw we lost the differences when they were grown in the same environment. So this one's all phenotypic plasticity. We changed environments, we changed the phenotype, but our populations became similar. This one, our populations remained different, and when we changed environments, they stayed different, they didn't change. In this one, we've got both our genetic contributions. Tall plants still remain taller than the short plants, and we had phenotypic plasticity. When we switched the environment, it caused a change in our phenotypes. All right. That's our common garden experiments. In this case, a reciprocal transfer study. It's useful. And so I've got the example, see the board, we laid this out, and that's our question that we were trying to address. Is the difference in our heights due to the genetics or to the phenotypic plasticity? And depends on what our graph looks like, gives us our answer. All right, ready? All right, so that takes some time, but it gets at phenotypic plasticity versus genetic contributions. Sometimes we don't necessarily care about that. Instead, maybe we are concerned about differentiation, maybe interested in ecotypes. Do we have separate ecotypes there? All right, do we have barriers to gene flow? Sometimes we look at our populations and they look identical, but we know that they're separated and they've been separated for a while, so maybe we're interested in how different are they genetically. About the only way to do this is through genetic analysis. Sequence the DNA and then compare similarity between our populations. All right. Gives us a lot of information. The downside is that this genetic data doesn't really reveal anything about adaptive variation, meaning that variation that could be under selection. Right? It just tells us basic information. Are they similar or not? Or how different are they? Doesn't tell us anything about, does that relate to a trait that selection can act on? Is this a trait that can be passed down through generations, so it's heritable? Doesn't really tell us that. All right, so we get our sequence, we compare them, and we can generate a similarity index called Wright's F statistic. Wright's F statistic tells us about the degree of similarity between our populations. This F statistic is very similar to the F statistic in an ANOVA. So those of you that have had stats, it's kind of where he derived it. So what is this? What is this Wright's F statistic? This statistic, Wright's F, is going to partition our total genetic variation, all that variation across our species, into variation between the populations and variation within a population. All right, so you've got this total genetic variation across the species. That total genetic variation can be broken up into the variation that it just occurs within a population itself, and it's the variation that exists between our populations. So we've got a couple different F values. So FIT is total genetic variation. FIS is the variation within the population. That's how, within any one of these populations, how different are they? Right? How different are individuals? And then we look at the average populations. How different are the individual populations for, let's say, single phenotype, or in this case, genetic similarity. The FST is our Wright's F statistic. All right, the FST is Wright's F statistic. What are the values? Well, we're not going to get into specific values. We'll just talk about large values versus small values. So what does a large FST value mean? If we have a large FST value, let's say a large FST relative to the FIS, then most of our variation, most of our genetic variation is due to differences between our populations. Yeah, we've got some variation within a population, but most of the difference across our species is due to differences between the populations themselves. Example that I'll use, and that I use in stats are these. We don't have a huge number of roaches. We got these black bugs walking all over the place now. All right, if we measure the length of those black bugs, and we've got the lengths of the black bugs that were captured in Cavness versus Science 3 versus the academic building. All right, we've got variation in lengths within each of the buildings. If we have a large FST value, then what that means is that the building lengths differ. Cavness may be the bigger ones, and Science 3 may be the smallest ones. Right? And what would produce that? What would produce these large FST values? Well, isolated populations. That's one thing. So if we're isolated, we have limited gene flow. If you have limited gene flow, then you're allowing evolution to take each population down its own evolutionary path. Second thing that could produce this large FST value are different environments with strong selection pressures in each. So these are three different environments. Each environment has its own set of selective pressures, and they're actually exerting pretty strong pressure, which is forcing these populations to evolve to their own environments. So again, we've taken our own evolutionary path. And then the other thing is our small population sizes. And you can say, well, that seems surprising, but it's not really surprising, because if we have small population sizes, then we allow drift to happen. All right? Drift becomes more effective in these small populations. So if we've got three different populations and they're small, drift is going to take each population down its own path. Those are the large FST values. What about small FST values? Well, it's the complete opposite. So with small FST values relative to the FIS, so small FST relative to the FIS, it means most of the variation in our population is existing within the individual populations themselves. Just say individual variation. Our populations are similar. That's what a small FST is telling us. So what can produce similar populations? Well, no barriers to gene flow. Anytime we start going down our own evolutionary path, gene flow wipes out that new variation or homogenizes the species. Similar environments with similar selection pressures. Instead of each population going down their own path, they're all basically traveling along the same course through evolutionary time. And then large populations. If these populations started similar to begin with and we have a large population, then the effective drift will be minimal. Trying to see. It looks like I have an extra slide in here that's just a duplicate. Yep, that's all it was. All right. So how did we get these FST values? Again, sequence analysis. Costly. You need a fairly large sequence. You can't do it with 100 base pairs. I mean, you can do it with 100 base pairs, but what good is that? You want to have several thousand base pairs to do this type of analysis. You want to try to get different genes to do these types of analysis. And it is kind of statistical. But the FST values tell us a lot about not only similarities, but about the processes that might make populations similar or different. And then once we know they're different, maybe then we start saying, okay, well, what's the cause? How much gene flow is there? What are the selective pressures? And so forth. Questions? This one? Or this one? Right? This one? All right. I'll make this, so this part was pretty easy. When we talk about quantifying, we've got phenotypes, common garden experiments, and we have genetic sequencing. Remember, downside of genetic sequencing, it doesn't tell us anything about adaptive variation. We just know they're different. And then we kind of have to start doing more digging to figure out what are those genes? Are they under selective pressures or not? All right. We just finished talking about Wright's F statistics, so know their interpretation. If we have a large FST relative to the FIS, we've got genetically different populations. That would be a case where we may have ecotypes. If we have small FST values relative to the FIS, our populations are similar. Maybe our distinction, or one more, maybe our distinction of these separate populations is wrong. Maybe they're not actually separate populations. Maybe they are one big population because the amount of gene flow homogenized. All right. Leave that up there. So this was a pretty short presentation. I'll have the quiz go live today, and you'll have until Friday to do it, end of the day on Friday. I think this quiz, based on the length, you may have like under 10 minutes to do it because it may only be 4 or 5 questions. All right. I don't remember it being very, very long. Not like the last one where we had a pretty long presentation. All right. All right. Move on. I know I told my lab this, but what we were doing was getting to populations. So we kind of finished up the evolutionary ecology section. And that really was evolutionary ecology. We talked about physiological ecology and put it in terms of evolution. Now, we're going to move towards the populations, population ecology. And when we talk about populations or when we study populations, we've got 4 basic descriptors. And these descriptors are demography. That's what's grouped in the demographics. All right. So demography is the quantitative description of a population and its characteristics. quantitative description of a population and its characteristics. Demographics, anyone that's interested in demography is interested in how populations grow or change through time. So grow and decline. How do they grow and decline, and how do they change? How does maybe their age class change? Or their age structure change? How does their sex ratios change? How does their spatial dispersion pattern change? And that's all demography. It's all demographics. Now we're at population. Now we're studying a population, so now it's kind of a good idea to really define what a population is. We define population as a group of individuals of the same species that inhabit a particular area. Right? The individuals of the same species are called conspecifics. So you may see definition as a group of conspecifics that inhabit a particular area. That just means it's a group of individuals of the same species. This definition is specific in defining same species and same area, because all of our individuals in a population must be able to interact with each other. They have to be able to interact. Why? Why is that? Reproduction. Right? This goes back to our evolution, our Hardy-Weinberg. All individuals have to be able to interact, and usually the way in which we talk about it is they interact through random mating. Now, just because they interact doesn't necessarily mean they're going to take part in random mating, because again, not all individuals in our population are at reproductive age or at sexual maturity. Yet they can still interact. Now, we recognize that our populations, that we can have multiple populations through larger areas. Our different populations are connected by dispersal. That's our gene flow. Connectivity describes that link between our populations. Right, so connectivity is defined as the link between two or more populations by dispersal of individuals. That's our gene flow. So you could say a link between two or more populations by gene flow. If we have high connectivity, we have more movement between our populations. If we have low connectivity, we have low amount of movement between our populations. So in general, if we have two populations with a high degree of connectivity, then those two populations are going to be more similar than two populations that have low connectivity. So the question is, if you have high connectivity between populations, what's our value, what's our FST value? Is it going to be large or is it going to be small? Small. Low gene flow, or I'm sorry, high gene flow homogenizes the genetics. So we should have high degree of genetic similarity, which gives us a small FST value. Ready? Almost. All right, so when dealing with populations, it's important to define our boundaries of what we're going to define populations, because our geography isn't homogeneous. We have spatial variation. We talked about what produces some of that spatial variation. And because of the spatial variation, not all individuals are going to be distributed evenly. You're going to see basically groups or individuals individuals of the species prefer certain areas over others, and that's where we find them, and so forth. All right, so in this example, this could be an area where we sampled for, let's say, mice, something small. All right? We have to be able to separate the mice that are found in the old growth forest versus the young forest and the forest patch and the old growth forest. And we just looking at this geography of it, do we think mice are going to be moving across that river? Probably not. All right. Are they going to be able to traverse the ravine? Probably not. Could they move to the forest patch? Maybe. Possibility. I mean, they have to get through the agricultural fields. All right. We assume that they live in the forests. So geographically, they look different. And we might be able to rationalize defining 4 different populations: an old growth forest population on this side of the river, an old growth forest population on the south of the river, young forest population, and then a forest patch population. You might ask, why? Why go through that trouble? Why not just say we've got populations north of the river and populations south of the river? Well, it's because this variation, how we define our populations, could have an impact on the important demographic measures, including number of individuals that are present or density. It could affect our spatial distribution, could affect our reproductive rates, could affect the degree of connectivity. It could affect other things, including growth rates. When you mature, how many offspring you have, how much parental investment do you have? All of these things could be influenced by our boundaries. All right, so that part of defining a population in a specific habitat, we have to be kind of specific about that. We have to be specific about that. And this graph kind of demonstrates it. So we look at our numbers. Yeah, the forest patch could have higher mice in it. Old growth forest, less. Does that mean necessarily that the mice do worse in the old growth forest? No, it could be a part of their demographics. Could be a part of their demographics and what they've been adapted to. All right, so this gets us to ecological versus evolutionary populations. And this is where we're talking about interaction versus random mating. All right, so the ecological population is the group of individuals of a species that occupy a defined area, particular habitat. All of our individuals need to interact in some way. The boundaries of these populations are usually determined by ecologically relevant changes in the environment. That's our spatial variation, our geographic variation. Evolutionary populations only consist of those individuals that are randomly mating. All right, so those group of individuals that randomly mate is called a deme. And the deme is what defines our evolutionary population. Our boundaries in this case are no longer defined by some ecological factor. They're not defined, they don't have to be defined by geographical variation. Rather, they're defined by the barriers to gene flow, barriers to mating. All right, so we've got kind of 2 different terms, 2 different types of population: ecological versus evolutionary. Evolution acts on that evolutionary population. It acts on the deme. All right, we already said evolution doesn't act on the individual, it acts on the group. And the group that evolution acts on is the deme, the group of individuals that are randomly mating. Now what's interesting is that if we have a change in our demes. So if evolution acts on the demes, then it will affect our ecological population. So this might seem a little confusing, but let's see if we can clarify it. Let's assume we are looking at tortoises. This. Say we're looking at tortoises. They inhabit an island. So here's our island. When we go out and look, we have a group of tortoises that inhabit that area of the island, a group of tortoises that inhabit this area of the island. We've got a group of tortoises that inhabit there. And then we have one A and this B. A, B, C, and D. All right, A, B, C, and D. The island itself could be the ecological population. So we could be justified in saying all of the individuals on this island constitute a population. Could they interact with each other? Maybe. Maybe. They could go out, they could interact. If they go out and feed between areas, you could say, well, B doesn't interact with D. Well, maybe it could. B can interact with C, which then interacts with D. Kind of an indirect type of interaction. But we've defined our area as the island, and that could be our ecological population. But then when we go in and start doing genetic sequences and start looking at similarities, we start seeing that we have 4 different groups on this area, on this island. So it's enough to where we can classify these A, B, C, and D groups as evolutionary populations, or as our demes, because these are the individuals that are randomly mating with each other. B doesn't necessarily randomly mate with D. B doesn't randomly mate with A. A doesn't randomly mate with C. So we've got our demes set up. Each of these would be a deme, but the island itself represents our ecological population. If we started to increase the connectivity between these, then maybe the entire island represents both an ecological population and an evolutionary population. It's entirely possible that that could happen. All right, I have a question. What if, as a rule, species does not mate randomly? Females are choosy, but the evolutionary definition is there just no population. So we recognize that female choice represents random mating, because it depends on her actually finding or being exposed to the mate. So her actual #1 preference, she might never ever encounter. That's our aspect of random mating. We have selection that could go on, the choosy part. All right, so that's our selective force, but that could be random mating. So hopefully that kind of answers your question. All right. So we have some fundamental descriptors of our population. I'm going to introduce these today before we end. So the first fundamental descriptor is the size of our population. How many individuals are there? In some cases, we report it as a density. Doesn't change the fact that we're reporting a size, population size. So that's a fundamental descriptor of a population, number of individuals. Number two, spatial dispersion. So number of individuals, that was the focus of last week and this week's lab. Spatial dispersion is the focus of our next two labs. And that is our spatial distribution across the landscape. Are we randomly distributed? Are we clumped or aggregated? Or are we uniformly distributed? That's our fundamental descriptor, number two. And then the third thing is demographic characteristics. This would be our age structure, sex ratio, birth rates by age, mortality rates, growth rates, age of maturity, parental care, all of those things represent our demographic characteristics. And what we're going to be doing is talking about each of these descriptors. And I think what we'll do is we'll start those on Friday. All right, so quiz should be live, hopefully. I said it was. I should have said it. 8 o'clock today, so choose the answer. You got 3 choices. The correct answer is 3107. (3107). That's our answer for the attendance quiz. All right, 3107. All right. If you have that, then we are free to leave. See y'all on Friday.